Hello, everyone. This is Rachel Heilman, the president and co-founder of the Rory Belle Foundation. I'm providing a recording update since I only recorded on one software program and not on both. This is recording from the April 17th family meeting and Genetics 101 update. You will see a separate recording for Sam Miller, who is a genetic counseling student from UPenn that we've partnered with for the last few months. He has a Genetics 101 informative recording session just on basis of genetics, and then also NARS1 and symptoms and things that are specific to the NARS1 disorder. So I'm going to provide an update here for all of the research and fundraising activities and everything else that the foundation has been doing for, gosh, almost the last year now, since the last time we met. So with that, I'm going to go ahead and get started. And if anybody actually needs the translation, please feel free to outreach me and I will download that in your language. Thank you so much. All right, here we go. Okay, and then for those of you who don't know me, I started the Rory Belle Foundation with Terry DeClerc. Both of our children are affected by NARS1 disorder, and we've established in 2022 and have been rolling right along since then. So here's our updates for really since the last time we met. Sam actually will cover quite a bit of this in his presentation in an easier to follow, digestible manner. But I think, again, what I wanted to just point out was, how many variants that we have across the NARS1 gene, including especially in this catalytic domain, and then in this UNE, which is very unique to mammals and humans. And then finally, this is actually very important because this helps us try to think about how we can treat NARS1 and find therapeutics and medicines for that disorder. And so we do have a repetitive or redundant variant, this arginine 534 nonsense, which many of you on the call are quite familiar with, but we're starting to see new variants pop up all the time in our community. So this is the Rory Belle Foundation roadmap. I have created this really in an effort to help maintain progress and to track where we're going and how we're doing. When you have a small nonprofit like ours in a very, very small community like we do, we really are the ones driving the research and collaborating and raising the money to fund medicines. And so, you all have done a lot by raising awareness and, have some specific action items at the end of this that really can be helpful. But this is essentially everything that has to be done to really get to this rocket at the very end and get options for our kids. And I'll be talking about a lot of these opportunities and where funding has gone throughout the presentation. So first, I wanted to just highlight RareX patient data. So Sam touched a little bit about this, but RareX is our data collection platform. This is entered by you as caregivers. It is extraordinarily important. I can't emphasize how important this is. We only have about 30 published cases in the literature. Many of those families have not engaged with the Rory Belle Foundation or with researchers apart from that diagnosis. And so we really need as many individuals as possible to enroll in RareX. You can see we've really kind of slowly gauged and chugged along here and almost doubled our enrollment since last year. We are multinational. We are all over the world. know we have additional families in France, and so would highly encourage anybody in France to enroll if they can outside of this one. We are missing a handful of others here in the U.S. as well. But again, so important. This is what drives us. This is what we understand about our literature and about our disorder. As you can see, our kids are getting diagnosed earlier and earlier, which is really important for us because it helps us guide and determine, you know, how our kids are developing. But I cannot express how important it is to have all ages enroll, because this really does help us understand how kids with NRS1 live, and, how they develop over time, what they're capable of doing, if they continue to gain skills, if they lose skills, et cetera. And so kids like James and Joseph are really, leading the path for us in understanding those things. So what I really wanted to highlight is, yes, we have actually 17 families enrolled in RareX, but only 13 have actually responded. This means that we're missing 4 families who have enrolled in RareX, but have filled out 0 surveys. So we cannot start talking about, endpoints and what's important until we can get as many as possible. We've really identified probably at least 30 more families across the world who are impacted by NRS1 in the last couple of years. And so, again, we really need, as many as possible to enroll. This is the head-to-toe survey. It's very important to do it. It helps us understand, where kids are, in their developmental state, as well as what systems are impacted. And for something like this, like digestive, for example, we actually didn't even know about this in the literature until you all participated in these surveys. And so this is clearly a huge symptom that, almost all of our families are facing. And this can include things like constipation, chronic vomiting, things of that nature. And so again, it's just so important that we're all enrolling and keeping up with the surveys. I wanted to highlight here, the really important surveys. So the ones boxed in red are the ones that we really need to do to complete for NRS1. It is a little overwhelming when you get into the RareX platform, all the surveys that are out there. So head to toe, this is top to bottom, all the organ systems that are impacted. The digestive system for us, I think, is really important, and is highlighting some new symptoms that we weren't aware of. And then these right here, ORCA measure is actually about communication. We know our kids struggle with being able to communicate successfully throughout all of their interactions, sleep, eating, and then quality of life. And finally, the Vineland. And the Vineland, I will fully admit, is a brutal and hard and long survey. So please take time to do this in chunks. It probably will take you about 2 to 3 hours. But this is what is considered standard and is a part of a lot of clinical trials. And this is why it's so important that we're submitting this and we're understanding it, even though it is really, truthfully, unpleasant to actually complete. It is so incredibly important. And then I actually didn't highlight this, but brain and nervous system, because we know NRS1 really begins up here in the brain. And this is what impacts a lot of our other body systems. So that's RareX. I implore you as much as possible to complete that. New surveys are coming out in French, Portuguese, and Spanish. So it'll be great for folks to be able to fill those out in their own language. And then also the genetic reports. We only have 7 genetic reports. We'll talk about that later, uploaded. If you can't find out where to do that, please, please, please let one of us know. Myself, Ross, Kit, Terry, we're all willing to help you do that. RareX will now take genetic reports in English, Spanish, German, French, and Portuguese. So again, we really need to make sure we're doing that so that we can track and understand variants and how they're connected to the symptoms. All right, progress this far. So over the last year and a half since Terry and I launched in July of 2022, we've raised just over $200,000, which may seem like a lot, but this money goes fast and quick, and we really need to actually raise around that much every single year to continue to move progress forward. And so the next couple of slides, I've really broken down what we've been able to do and accomplish by teams. When we started this in 2022, and I'm sure most of you families are well aware, Stephanie FMU, who was part of the team that discovered NARS1 as causing a disorder, was the only researcher that had eyes on this. Right now, she has two sets of fibroblasts, which are skin cells that you can actually use to generate what's called an iPSC, which is essentially programmable cells. This means she can actually grow them into neurons, which is so important for us to understand how the brain develops. She has one case or one set of autosomal recessive or inherited, and then one set of this arginine 534 nonsense variant that were donated by families. This means that what she's doing is she's looking at all of us, right? So she's able to look at, our kids that are de novo who are coming, who are developing embryonically or, having a new variant, and then also our kids that are affected and inheriting variants from each of their parents. So she continues to remain the sort of central collection for new cases and is working on those neurons. Next, we have a mouse team. One of the things that Stephanie was able to do as part of her doctorate was to develop an autosomal recessive or an inherited mouse with Harwell Labs and Jane Slay in London. This mouse looks like our kids. So these mice have seizures. They walk a little off balance and drag a leg. It is quite impressive. And it's so important that we can see that in an animal, because then as we look for medicines, we look to improve those symptoms. One of the things that we've been able to do in the last year is collaborate with Jackson Labs, which is located here in the United States, and apply through an NIH grant to get a mouse for our de novo cases. Again, trying to envelop or incorporate all of the kids that we can in the work that we're doing. So this mouse model is underway. We will definitely need funding to phenotype, which means we're to assess this mouse for it to look like our kids that are impacted with these de novo or new variants. That will likely cost us between $30,000 and $50,000 to do, and we'll need to do it in the next year or so. Our other team is what I'm calling our yeast team. So NARS1 is so incredibly important. It is found in as simple as a life form as yeast, all the way up through humans. And so we contracted with Perella Labs here in the United States. What they do is they remove the dead 81 gene, which is essentially the NARS1 equivalent gene in yeast, and they watch that yeast grow. And so we've already heard back. It's been about a month that our yeast are growing slow. There's going to be a press release from this soon. This is really great news. We want our yeast to grow slow. We want it to look bad. Because what they do next is they essentially take 7,000 different drugs that are already available, either here in the US or around the world, or over-the-counter supplements, and they put that on different in little cells, little dishes of yeast that are growing slow, and they look for that yeast to change. They look for it to grow better, and that actually will generate us a what we would call a hit list, or a list of medications that we hope will be safe and effective and available, that we can try for our kids. And again, this is for, as many kids as we can possibly, impact, de novo and inherited. We funded this. This will cost us about $72,000. We're hoping that with my background and Kit or Catherine's background as pharmacists, this will help lessen the impact of some of the costs down the road, just because we can very easily look at medications, since we have a pharmacy background. And then as a follow-up to that, we've been able to meet a lovely researcher in Canada when we went for a large conference on the R's genes, Ilka Heinemann. She actually just hired a postdoc to create yeast models of every known individual with variants. So if you've been in Rare X and you've been able to pull variants, for those of you who have, I've consented for our repository here. We've asked her to build models of yeast that look like your children. And again, what we should see is those yeast will grow slow, or will grow maybe erratic, or won't be the right shape. Our goal then is to test the drugs that we get from Perella Labs on those yeast models and look for correcting it, look for safety, make sure that we're not worsening the growth of the yeast, but also making sure that it's improving. So it's complicated. But again, this is, really for us something that we want to do to make sure we are able to find drugs that will help, as many of the kids affected by NARS1 as possible. She was able to apply for a small grant in Canada with some local clinical support, but will likely need some additional funding for this work since she's building our individual models. So next actually is something called tRNA run-through, and that team, and that's Patrick O'Donoghue, also at Western University. This is actually Ilka's husband. So we met both of them. Terry and I met both of them last year through the Rs conference in Canada. tRNA run-through is a relatively new technology, but has a lot of promise. So there's already a handful of biotech companies and pharmaceutical companies testing this in what we call preclinical, so no therapeutic options right now out on the market. But there's some mouse models that are showing promise in other diseases, not NARS1. But we actually have been able to get those fibroblasts or those skin cells up there to his lab. And essentially what they'll do is they can essentially add this arginine or this ARG right here to the gene, or to a system, and as it attaches to the gene, it reads through that. And so instead of it stopping like what's happening right now in kids with this disorder, it'll read right through there. So we're really excited about this. This has a lot of promise. And we hope that, we'll be able to expand on this, and provide updates. So this is really, really exciting, especially for this population of our kids that have this very particular mutation. To follow up on that, we have what we call this arginine 534 nonsense functional team. So anytime you have a change or a variant in your genes and in that protein, it can impact that protein differently. And so there's a doctorate candidate in Zhang Li at Scripps Lab here in California. And what they're saying is they are finding this to be dominant negative. And what that means is, you know, what the choices that we choose for medicines or for drugs for this particular variant, which actually impacts a large population, is going to be different than what we use potentially for other choices. And so this is where like tRNA run through comes into play, ASOs or oligonucleotides, which is a well-established therapeutic option. So as we figure these things out, it helps guide choices down the road. And then finally, we have a thermal mechanism team. Thermal just means temperature. So Marissa Seamus was also here at the ARS conference last summer, who we met. Their lab's based in the Netherlands. They also developed this amino acid assay, which is hugely important for us to figure out how well NARS1 functions. And if there's less enzyme, then we also know we're not adding enough asparagine to the end. So this lab and Marissa is very important. She's been testing this thermal dysfunction or temperature sensitivity on both autosomal recessive, so inherited cells, as well as this arginine 534 variant cells. Terry, myself, Ross, Catherine, we're all getting ready to meet with them soon to kind of talk about next steps there. But essentially, what you might see in your kids, if you have what we would call a positive thermal reaction, is when your kids get sick or spike a fever, they have a harder time recovering. Their skills, they might regress, right? Their language, they might regress. And it just kind of hangs on. That illness hangs on for a while, right? Their ability to regain the skills or to kind of get back to their baseline is a little bit more challenging. They might be more likely to have seizures during this time period if they do. Or just a lot harder to recover. That's how it presents in humans. She right now is testing this in cells. This says they have seen this in other RS genes, so that cluster that NARS1 belongs to. And so we're really hoping to get some other medicines that could help prevent this or prevent, when your kid becomes ill from that happening. So we're excited about this opportunity as well. All right, goals for 2024. So these are things that as a board we're really talking about. How do we get there? Things that we've already done, things that we need to really fill those gaps on that roadmap that I showed you earlier. These are really coming from us. We are funding these things with the money that we've raised, and with the money that the James Gang has raised and donated to us, or that Ross has done, or Kat is doing right now. So I think again, this just kind of goes to show, right, how much we need everybody to support, moving this effort forward for all of your kids. So cell line growth. So cells are incredibly important in us understanding what's happening in the body. iPSC cells, we've been able to collect quite a few of these. Again, this can help us grow neurons, which is so important for us to understand. We have one in process right now and two more that we've hopefully collecting here in the next couple of weeks, as well as a small skin sample. This is all here in the US. It's very hard for us to collect abroad, but I certainly have helped Stephanie collect and certainly am able to do that if we can. Scientific research. So a lot of this stuff is pending, but already funded. One is metabolomics. This is something that industry funded here in Combined Brain, which is a nonprofit I'm associated with. This will help us see like what's happening in our kids' bodies in terms of sort of metabolites or breakdown. Somologic is proteomics or protein. So we know that NARS1 is a very important protein. and proteins interact with other proteins and tell them what to do or what not to do. And so these can be really important units of measurement and help us understand where things are not working properly in kids with NARS1. So we've been able to collect about 4 or 5 different kids here in the US of kids with NARS1 and also their unaffected siblings. So this will be fairly expensive to run. I just found out actually it's probably going to be closer to $800 a sample. So we'll continue to kind of figure out how we're going to have to fund those to get that really valuable and important information. And then we're just exploring simple animal models, so flies or frogs. Yep. We like all the animals. This will help us again understand like what does NARS1 do in the body. We do not actually fully understand that, but this will help us really target that it's efficient, it's fast, and relatively inexpensive in the grand scheme of things. So hopefully some really exciting funding that will happen there. And science overseas or in Europe that will help us with this near-term exploration. So one of the things that we're really focused on right now is this AAV9 vector design. So this is classic gene therapy. There are a handful of gene therapies that are available for disorders already approved on the market. This will work in our inherited cases or in other cases where there's an elimination of the function of NARS1. We have met with a handful of folks just to try to get a vector design to deliver the gene. It'll cost at least $100,000 for the first year. We're likely looking at $2 to $3 million just to kind of get a therapeutic together over the next handful of years. This will hopefully run in parallel with like tRNA run through or ASO therapeutics that will really help with the other sort of segment of our population. High throughput testing. So this is again going back to those yeast models that I mentioned, and that cost mouse characterizing I mentioned, and then one of our huge goals is really to get all of us together, or as many of us as that we can get together along with our researchers, either in London with Stephanie there at the UK, and we have a small group of families, or on the East Coast of the US. This will cost between $50,000 and $70,000 to set everything up. But it is something that is invaluable to, moving us forward as a foundation and also us together as a community and really bringing more and more attention to each of your children from scientists and clinicians around the world. Again, I just kind of wanted to show you some other things that we're really working on. So meeting attendance, and I know Kim in Australia was able to go to the local epilepsy meeting there. Ross and Rosalind and were able to attend Rare in England, and Ross was little boy was a rare difference maker, challenge maker. For this last rare disease. So again, this is so important. The more things that you guys can do to attend and bring attention to NARS1, the better off we are. There's only 60 of us, right? So just saying the name can change the direction of researchers and clinicians. So these are the things we've already done this year, and the things that we have planned. So there's a lot of travel and an investment here on our side to really, move this forward and bring attention to NARS1. And then fundraisers, hoping to do a virtual global 5K. I'm working on a wine tasting effort in Denver, and then Kit is helping launch an international merch store. We have Bonfire available, but this really will help keep the cost down and for all of our families in Europe and Australia, around the world, to really help be able to get a more affordable merchandise to, advertise your support, not just for the Rory Belle Foundation, but for advocate for your own kids. Um, so our ask to you, so this isn't just about what we're doing, but also, right, how you can help and what you can do for your kid. So RareX, like, please sign up if you haven't, right? The genetic reports, as I mentioned, there's only 7. Like, we have to have those because that tells us, any new variants and then the symptoms that are really associated with those variants. So we can really start to understand, how things are similar, how things are different. Head to toe surveys, incredibly important. And then these surveys listed here, to help us understand communication, behavior, sleep, seizures, etc. Happy to help in whatever way possible. What else can you do? We have a handful of families who have their own social media pages, following their kids. So please do that, or, submit your story through the Rory Belle website. I'll reach myself or Ross Terry to do that. Joining local rare disease organizations can make a huge difference. So doing the things that Ross and Kim Roslin have all done across the world, those can change the awareness, as well as attending meetings in your area. Fundraise. We will certainly need to continue to raise a lot of money to move this forward. So Facebook fundraisers, we use GiveSmart. We can set up personal links. We have the package to hold a gala, which we did last year. Terry did an amazing job with that. Any small event helps. We have a laundry list of volunteers. Thank you for those who have helped. We're always looking for more, and we will try to figure out really how to make that easy for you and to use your skills to move those things forward. That's all I have. Hopefully I answered a lot of questions in the chat from before. I'm so sorry I did not record this the way that I wanted to. Please message us at any time, post on Facebook. I know Ross isn't here to sort of plug the other things that he's doing as part of our new Director of Community Engagement, but he's building a new website. Kat is helping him do that as well. We're hoping to get more active on social media and make things a little bit more user-friendly. We were awarded a grant last year, and so Wordly is our translation service. We will work to continue to refine that and to work as much as possible to be able to bring these things to you in your language, so that you can understand and we can have better and more open lines of communication. So with that, thank you so much. I appreciate everybody's efforts in showing up today. Take care and don't hesitate to outreach.